Okay, how many of you used an external camera for either exam 1 or 2? 1, 2, 3. Yeah, it looks like about 25 to 30% as I go through and look at the ProctorU videos. That's what I'm finding too, and it's taken a while to go through those. I'd like, if you did use a camera, I'd like to see the positioning so I can make recommendations for exam 2. Some of the camera situations, the Proctor didn't improve it much. Other people have got really good views. What I've found, just looking at it myself, off to the side instead of like that diagram, looking from the front because your computer gets in the way, off from the side, up a little bit on a stack of books, but ProctorU has this silly rule that you can't have any books. I would say you can't open any books. But in order to open a book, you've got to take the camera off. So one person had, their desk was a board on stacks of books. Of course, the only way he could open those books would be to disrupt his entire desk and everything, but ProctorU made him put a blanket over those closed books. So I think there's a lot of silliness going on with the proctoring arrangement. I've seen enough of these videos, and I see some of you seem to wait forever and ever, and I can see the frustration building in your face, and the ProctorU is like a robot talking, you know, do this. And then 5 minutes later, can you take yours in and move all around? Can you take a mirror? And you're going, yeah, I'm forgetting all this stuff I've been cramming for 2 days. So I certainly sympathize with you, and I feel like I really know a lot of you, and I know the rest of you because I've seen you under extreme duress. I see how you handle stress, about like I would handle stress. Okay, so today we're going to finish up recombination, but you know, I'm always giving these lectures to myself actually, and every year I go, I don't understand Rec A. And the book sort of says, you know, you get the one strand and then it scans a double-stranded duplex without melting it. One piece of DNA scans another piece of DNA to form synapses, and then the strands exchange. How does any of that happen? The current textbook doesn't give you much clue. It gives you these kind of very opaque diagrams. The details are not really known satisfactorily yet, but we know a lot more than those diagrams in the textbook. I went to molecular biology of the gene, which I'm apt to do on occasion, and I've put some figures in there from that. I'm not saying that that's a better textbook. For some things, though, it has more molecular detail in the explanation. So I found it enlightening. So this is from molecular biology of the gene, and the first item is, of course, you all know that Rec A has a specific direction in which it binds to DNA. The first one binds, and then the rest have a strong preference for extending this skeleton, this scaffold, from 5' towards 3', as if it was a transcript elongating. So you hit it here, and it'll go all the way and get to the very 3' terminus. So that's one thing that's sort of illustrated here. The other is this idea of how do you scan? How does a single strand scan a double strand? So here's the Rec A filament. You have 2 sites, 2 binding sites. One is called the primary site, the other is called the secondary site. The primary site is what is occupied initially by the single strand, the protruding single strand that's loaded by the Rec BC D complex. So that's what you see right here, the single strand. And then there's a secondary binding site that is not very high affinity, and it forms a very transient interaction. So it allows the double strand molecule to slide back and forth quite readily by interacting with this secondary binding site. Now, this appears to happen while the double-stranded DNA is on the outside of this big circular tube here, this winding serpentine tube. And so that part of the mechanism, for me at least, is unclear. In the diagrams you see, single strand is coded by the Rec-A, but they never show the triple strand structure, but yet there is X-ray crystallographic data showing it. But how do you transition from a single strand to a triple strand and then back where you lose a strand? There's still some sort of missing steps, but the other aspects that are informative here are the fact that the Rec-A monomer binds 3 nucleotides at a time. 3 nucleotides. Where have you heard that before? That's a codon. If it was RNA, it seems that even in the DNA structure, there's a natural grouping, kind of a repetitive grouping of 3 nucleotides in the B form of DNA. So that may underlie the fact we have 3 amino acids, or 3 nucleotides in our genetic code, stemming all the way back from the base pairing interactions of DNA. So Rec-A binds 3, they naturally lump together, and this is what it scans with. 3 at a time are scanning the double-stranded DNA without opening the double-stranded DNA. And so here's the double-stranded DNA here. One strand, the other strand, you're looking down the strands, see? The base pair would be like that, and you would open them up like that. So you take a slice and you look down the strand, one strand, the other strand. What happens is the single strand is interrogating these bonds, and it will displace this second strand, the one that's the same as it, because it needs to substitute for it to make this work. And here you see it now has formed the base pair. This happens just 3 nucleotides at a time. But if the one next to it also is able to do it, then you start getting a zippering action occur. So this is sort of the molecular detail of how this works, and you can see it's very precise. It has to be before this will work. This is not a very high probability event. Here you see the diagram showing Rec-A wrapped around single-stranded DNA, and here you see the heteroduplex. And that, they've drawn it on the outside, not on the inside. Is this an intermediate? I think it should be on the inside eventually, because if you look at the DNA, this is the trick. When Rec-A wraps around the single strand, it opens up and positions the bases as if they were in a duplex. If you just stripped away one strand, the strand that's remaining would be twisted. Rec-A mimics that configuration. And here you see it here. This is the duplex. You take away one strand and only a single strand is left, but notice it's in the same configuration it was originally. It's a little more stretched out. And then you see the heteroduplex forming. Notice in clusters of 3, mimicking the binding of Rec-A itself in clusters of 3, and then you see the displaced single strand now assumes a sort of a random configuration. So this Rec-A actually is a very precise positioning scaffold to get the single strand in a position as if it was already wrapped around its partner. And in that position, it's able to scan the closed duplex, as it's presented in a very transient sliding, as it slides past 3 nucleotides at a time, of course, along its entire length. So it's still a pretty miraculous event. Here you see the Rec A, each is a different color, as it begins to wrap around the single-stranded DNA. It's hard to see, but the dark red here in the center, that's that one strand. And notice it's not straight, it has the same configuration or conformation as if it was wrapped around its partner. And here you see in the middle of Rec A, the double-stranded final product, where this mauve strand has now invaded and base paired with it. So I think that, to me, is a step towards understanding it, a baby step from where I was before. And so, you know, this is not very informative, but this is kind of the level we were at before. And we'll go through these to get to where we left off. The RUV system. Recombination UV. The R for recombination, they irradiated cells with ultraviolet radiation, and a whole series of mutations occurred that interfered with recombination. And so the name of that category of mutations, which of course represent the underlying machinery, it turns out to be involved in branch migration, which is a part of recombination. So there are way too many slides on this. It's not that complicated. I'm going to edit it down and simplify. So I'll go through some of these pretty fast because they are kind of redundant. They built up over the years. We don't really need to spend that much time on it. But you do need to know all the players here. So you have RUV A, B, and C. RUV A and B are shown here. RUV B is a helicase, and there are 2 copies of it, RUV B. And this drives this branch migration. You know, the recombinant joint, this crossover configuration, it moves thousands of bases in either direction to displace and lay down the strand from the partner molecule. So branch migration occurs in most recombination, and this is the engine that drives that movement. These 2 helicases here, RUV B. RUV B is the engine. RUV A is a scaffold. It binds to the crossover point itself and facilitates a rearrangement so that the joint, just simple crossover, becomes the classic holiday structure. So let's proceed here. Here you see recombination happens at double-strand breaks, but it also happens sort of on small scale wherever you have a gap being repaired lots of times. So this is recombination, strand exchange occurring during a gap repair operation, and you get this recombinant joint formed here, and before it gets ligated, you get a movement of that crossover, that recombinant joint along the junction area. And then finally, the last step is RUV C is the endonuclease that resolves the structure even into either patch or crossover recombination. And here, of course, is the holiday structure. And this is formed by laying on the scaffold presented by RUV A. So B is the helicase, the motive force. A is the scaffold that spreads it all out on the table. And C is the endonuclease that cuts it. Here you see some more diagrams of it. RUV B, the motor, the scaffold in the middle is RUV A. go through this fairly quickly. And this slide seems to be out of order. I just noticed that before I came over it. This should be clustered with the Rec A slides. This is Rec A functionally in eukaryotes. You notice it looks nothing like the bacterial Rec A, but it does the same thing. I mean, if you think we don't know how Rec A works, we really don't know much about how the eukaryotic functional homolog works. And it's called DCM1 in humans, there, DCM1 in humans, and in archaea and yeast, it's called RAD51. Not to be confused with RAD50. There's a 50 there also that's involved in resection. RAD51 is the yeast homolog to Rec A. Human DMC1 is the name of the same complex in humans, in us. And notice it's a big double donut with the double-stranded DNA in the middle and regions for invasion of single strands coming in through the pores in this double donut. Pretty mysterious. Here you see a hypothetical model for how it assembles, but notice what's in the middle this time is double-stranded DNA, and then single-strand DNA gains access through the pores of this double donut. I don't know how it works. I don't think anybody really knows. Totally different in strategy, but accomplishes the same thing. Strand invasion. And then you have some of these little silly figures that don't explain a lot, but sort of summarize what's happening here, and we'll quickly go through these. The scaffold, RAV-A, creates a surface for forming the holiday, what's it called, holiday structure. The isomerization, if we get to a diagram, okay, this is, we'll get to it in a minute. This is RUV-C now, after it's all laid out. It's really presented this way so RUV-C can cut. RUV-C binds, it cuts at a particular site. I think it's ATTG or something. It has a particular site and it may have directionality into its site. So it doesn't cut totally randomly, but can either cut in this direction or in this direction, of course, with different outcomes. If it cuts, as you see here, then little a is joined, no, wait a minute, if it cuts there, little b is joined to big A. And large B, yeah, it's like that. It's so intuitive I can't do it. Here you see more structures. This is RUV-A forming the scaffold that the DNA is spread out on. And this is the mental gymnastics that you can end up with a sprained back if you try to do this. So I'm going to lead you through it, make sure you don't fall off the mat. Let's start, well, we'll start over here and then, you know, it naturally occurs the other way. This is the holiday structure, the final result. This is where you want to go. So how do you get from here to there? 2 steps. First is you take everything from here all the way like that. Just leave that part like it is and just rotate that around so this red is straight across, see? And then the green and the blue now comes up. Rotate it 90 degrees and you get that. Whew, that was work. Now, all you need to do is rotate that around this vertical axis. Take that and rotate it here, so now it's over here, and there you go. That can hurt if you don't have somebody there with you to catch you when you end up doing that. Especially on exam, people, I won't ask you to do that on exam because I don't want the insurance liability. And more of the same. I must really like this stuff. Oh, I don't know whether I'm going forward or backwards. We finally got through that rough spell, rough ABC. Now we're going to summarize a little bit. Notice the speed. That's not really fast compared to DNA replication, is it? So it's 10 to 20 base pairs per second, kind of a leisurely stroll. But notice it's in the process, if there's any rec A left there, it gets bumped off by this process. There's another helicase here that can participate, rec G. It can assume the role of RUV-B if RUV-B is the primary engine, but if this is mutated, you can get the same function fulfilled by rec G. That's the site for cut, ATTG. Mammals have a similar complex in yeast. It's MUS81. And that's the end of recombination. I don't see any tears, so I'll go on. We'll try to recover, get our emotions back in check. Recombination. Notice there's sort of a common theme here. DNA replication is often a part. It's sort of, these all sort of start as a variation of DNA replication, and that, of course, is the common thread among these slides. And the next part here is, if I can't see very well, there we go. Transposition, transposons. Transposons in some eukaryotes are a minor component of the genome. In us, over 50% of all the DNA we inherit from our mothers and fathers and grandmothers and grandfathers is total junk. It's leftover things that are dangerous, transposons. Transposons transpose. They jump, they move from place to place, and when that happens, it can create all sorts of havoc. It can generate cancer, it can cause deletions, insertions, a loss of chromosome arms. It's bad news. And yet, over 50% of our genome is comprised of things that have potential to really destroy us as a species. Transposons. How did they get there? What purpose? Why are they still there if they're so harmful? Why weren't they selected against? Because they're so dangerous, there are elaborate mechanisms in place to make sure they never escape, except it's not foolproof. And we'll talk about some of these mechanisms. Epigenetics and silencing RNAs and things like that seem to be part of that general strategy to keep these large portions of the genome from ever seeing the light of day of expression. 50%. How much of the human genome actually encodes for a protein? It's 1.5%. Of course, there are structural RNAs. The ribosomal RNAs, tRNAs, are structural RNAs, but still, the amount of sequence in exons, after you've trimmed away all the introns and everything, it's only 2% of the genome actually codes for the 28 to 30,000 genes that we have. Okay, I digress. Transposable elements, what are they? Well, jumping genes. Transposition is an integral part of their existence. There are two general strategies for jumping. One is you jump and you leave a big hole where you left the donor. And you just care about yourself. You jump and you go, sorry, look what I did, but I'm in brand new DNA, so everybody's okay. Never look behind, just throw that rear view mirror away. And the poor DNA left behind's going, what just happened here? I got a big hole. Double-stranded breaks, recombination has to repair that. Non-homologous, wouldn't you say? So that's one kind. The other kind is when it jumps, it gives birth to itself. It leaves a copy of itself behind, and it jumps to a new location, and it's going, my tribe increases. More havoc. Bacterial transposons. Well, there are two other ways you can categorize them. Transposons that are made solely of DNA transposons, and those which have an RNA stage, where they have to have make DNA from RNA in kind of a retrograde action. Retro RNA kind of goes backwards. So let's begin here. Bacterial transposons, carry that transpose themselves, similar to ones in eukaryotes, is really the same thing in us as in bacteria. Although many are defective, they can rely on enzymes from a few that are still functional. What does that mean? You get many copies of the transposon. Every transposon originally had at least one open reading frame. It encodes one enzyme, the transposase itself, that does the cutting and attacking and ligation, the transposase. But sometimes, if you have many, many copies of the transposon, a lot of them get lazy, they get mutations, but who cares? Because they still have the cut sequences, sort of like ORI-T. They lose their functional transposase, but as long as somebody's got a good copy, they don't seem to care. Move me. Make me jump. I can't do it myself. Give me a little boost, Seb. And they kind of sponge on all the ones that are still unmutated. And so many and most have mutated away from a functional transposon, but they still have these cut sites. They can still replicate, increase their numbers. Transpositions, we mentioned, can do all sorts of bad things. Can cause deletions, inversions, lead to movement of host sequences to new locations. They can serve as substrates for recombination, whether they function or not. In bacteria especially, you have two transposons. They are the same, in the same family. They have a lot of sequence homology. E. coli just recombines them, just without thinking. You know, that's how F plasmid and various plasmids get integrated into the genome. It's not anything encoded by the plasmid. It just happens because they're infected with transposons, and the host is infected by the same transposon, so it's homologous recombination. They're often called selfish DNA, but you know, all DNA is selfish. It just wants to make more of itself. Sometimes it robes itself with the magnificent garb of an emperor. That's a eukaryotic organism. Other times it's quite humble. It just carries its little bag of transposase. Selfish DNA. Transposons, genetic elements that are mobile. A major cause of variation in almost all genomes are caused by transposable elements, and their presence can have a major impact on the overall size of the genome. I don't know why I thought that was so profound, but it's in quotes. In maize and humans, roughly half the genome is occupied by these little things. Now we're going to get into briefly into how you can organize in your head, how to think about them. The classes of transposons, you see there are two general classes. They're, of course, listed backwards from the way you and I would list these things. You've got the retro element as class 1, and the very primitive early one that's just based on DNA as class 2. Retro elements which have an RNA stage and the standard DNA type element that has no RNA step. We'll talk about the second class first because it's the original and simplest. Directly propagate as DNA, jump into new sites, and they encode, at a minimum, a transposase. Some of them carry 2 proteins. Those that jump and make a copy of themselves end up with an intermediate that forms a co-integrant. And they have to have a way to resolve themselves. The original DNA, the donor and target DNAs have to resolve by recombination, so they're now 2 separate entities again. So they have an enzyme called, strangely enough, resolvase. Transposase to jump, resolvase to get rid of the co-integrant. The resolvase is a site-specific recombinase. It has a particular site that it works at only, called RES for resolve. Question 18, I believe, exam 2. Years 1856 through 2017, I believe. The DNA type, of course, are common both in bacteria and in us. We have the DNA type transposon. You know, Indian corn, it's called, the one with the colorful decorations. That's due to mobility of a DNA-based transposon. The natural unmutated color of corn cob, corn kernels, is kind of a bronze purple. When it has a transposon that jumps in the anthrocyan and pigment gene, it mutates it and it's this sickly yellow. We love the mutant. How about that other class? Has an RNA intermediate. You have 2 general forms. You have those, this is GoPro likes to send me a text halfway through. Yes, I'm trying to get through this. I'll be there in a minute. It's gotten so like it's waiting for me at home. It wants me to go to bed early. It's just like it's taking over. There are 2 classes of the retro elements. We call elements because they have 2 different names within them. The long terminal repeat retrotransposons and the non-long terminal repeat retrotransposons. The bottom class is sometimes called the poly-A class. So let's look at the LTR class. It itself is divided into 2. The first is no infectious particle. It forms a particle, but it doesn't infect anybody but itself. And we'll see an example of that. Also, exam question 22. You'll be able to look at that and go, that's easy. And the other type, which we're more familiar with, retroviruses. Okay, I'm trying to speed it up. Retroviruses, closely related, they form a coat protein, except this coat protein is secreted, this particle is secreted and infects other cells. A retrovirus. HIV is a retrovirus. And then you have these down here that don't have any kind of long terminal repeat at all. And we'll just briefly talk about these. They have a very unique and strange, uncomplicated, and poorly understood, paradoxically, way to replicate. Class 2. This is what we're going to delve into first, and then at the end we'll look at the retros. So these are the DNA-based ones. These elements don't have any free form. They're always integrated into either a plasmid or even a phage or a host genome. No relationship between its sequences and its target. It can jump any place. It is not looking for homology. only transport their own sequences internally within the genome. They don't pick up stuff, except we'll see sort of an exception called a composite transposons. So it's just a sequence that codes for a transposase. It's like an enzyme that has its gene, and the enzyme and its gene just visit different places in the genome throughout their history. Just having fun, just a little T here, and then they jump someplace else and visit. But they don't infect anything else, except if that genomic DNA is carried to another organism, like through bacterial conjugation, for example. Transposition generates a new copy at a random site. Well, sometimes it creates a new copy. Certainly there's a copy at a random site on a recipient DNA. Sometimes it leaves a copy of itself. Other times it leaves just a big hole. These are some of the consequences. If you have them on two chromosomal arms, you get recombination and you get rearrangements, large-scale rearrangements, unequal crossing over. Retroviruses are related to the LTR retrotransposons. These are the ones that are not infective. But here you see just the general outline of how retroviruses work. They start with an RNA that is then reverse transcribed by reverse transcriptase, encoded by the retrovirus, into linear DNA, and that linear DNA is then integrated, and then that integrated retrovirus is then transcribed by RNA polymerase II, the eukaryotic workhorse for protein encoding genes, back to RNA again, and it continues this cycle. The LTR retrotransposons make this infectious particle, but it remains inside the cell. It's in the cytoplasm, and it infects its own nucleus. Isn't that a shame? The retroviruses are made up, the particle is made of the membrane itself with some spikes, and it gets pinched off, and now it can infect a second cell. DNA type 2 elements, they hop. These are figures from the book. No homology required. Okay, so let's start at the most simple of forms, the insertion sequence. The insertion sequence is a transposon. It's a DNA-based transposon. It's as simple as you can get. So we need to know the components of it. What you'll see is the IS elements are named with IS and then a number. Notice that when they insert, there's some repeats involved here. So this is important that you understand the nature of these repeats and how they work. Notice the transposon here is in pink. And they've signified the repeat just by writing the numbers reversed here. 1 through 9, then 9 through 1. This would be an inverted repeat. The transposons at their ends have an inverted repeat. It's not very long, but here you see it. 9 bases is quite common. When they jump, all of the pink part goes, whether they leave anything behind or not. These repeats actually form the site that the transposase recognizes as the cut site. That's why these have to be without mutation. Otherwise, the transposase itself will never cut them. As long as you have intact inverted repeats, this thing can keep moving whether or not the internal open reading frame for the transposase has been mutated or not. So there's another repeat here. Notice in the target, It has no sequence homology with the transposon, but in the target, as a consequence of the jump, of the insertion, you get formed a direct repeat. ATGCA, ATGCA, at the target site. Direct repeat. That's sort of the signature that what's in between is probably a transposon. That's present in bacteria and eukaryotes, and it stems from the repair operation that has to occur, because when the transposon cuts to insert itself, it doesn't make a blunt end. It makes a staggered cut. And so as you take the top away from the bottom and you repair it on both sides, you end up duplicating that little stagger part. So the length of the direct repeat is directly related to how much the cut is staggered. If there was no stagger, there'd be no repeat. That's, I think, a homework and exam question. Directly related to the architecture of the cutting enzyme. How far the two cut active sites are spaced from each other. Does it cut 9 bases apart, 7 bases apart? Determines in that repair operation how large the direct repeat will be at the target. So here you see the target repeat. This is that direct repeat. A lot of times it's 9, but it doesn't have to be 9. The inverted repeats, which are, of course, inside the transposon itself, are a little bit larger. Notice the overall length of the transposons are about 1,500 bases, 1.5 kilobases. That's an average size to encode one enzyme. So an IS element is simply inverted repeats that are on either side of a transposase. Of course, the inverted repeats have to be recognized by that particular transposase. The different classes of transposase, they recognize a different cut site. And here you have one of these ancient, probably get edited out. So the inverted repeats are part of the transposon itself. They are carried, and it generates direct repeats at the target. Smallest transposons. Okay, here you see the cut. It's staggered. In this case, the direct repeat that would be generated as you repaired it on both strands would be 3 bases. That's the length of the offset of the stagger. So direct repeat generated simply by the gap repair because it's a staggered cut. I think on the exam, maybe it's on the homework or both, I don't know, but there's a question where I ask you to tell me what the repeat would look like. And the way you do that, I'll give you the staggered cut and you go, well, what does it take to repair? So the amount the nucleotides that have to be put in to repair would be those that would be found in the direct repeat. Most bacteria contain several of these IS elements. Oh, here you see an IS element. It has open reading frame A and open reading frame B. So this is an example where the IS element contains 2 genes. You just look at this and you know this is going to be a replicative transposition because it's has a resolvase. It has transposase, and the second gene is always resolvase. They always have transposase, and if they make a copy of themselves when they jump, they need a way to resolve the co-integrant, so the second one will be the resolvase. Here you see the repair generating the direct repeat. So it looks like okay, direct repeats. How many times do I need to get this across? Probably less than I have. Okay. So if you had a culture of bacteria and you saw mutations happening at a certain frequency, would you know it was a transposition, a transposon waking up and moving around? Or would you think maybe it's just random mutation, maybe there's some radiation or some kind of chemical mutagen about? Could you tell by frequency? The answer is no. They have roughly the same occurrence. When you get a mutation because of transposons, it jumped and inserted itself into a second gene. This is in bacteria. It's about the same frequency with which you would expect a natural mutation to occur. So you couldn't tell by that alone. You can see that the rate of spontaneous mutation is 10 to the minus 5 to 10 to the minus 7. Transposition is a little more frequent. A little more frequent. So just by chance alone, you got a transposition event. But it's very hard to tell. How about reversion? And this means when you have a non-replicative transposition, it leaves this hole. Well, what's that hole got? It's got direct repeat. So it's got 9 bases inserted, right? Or maybe it's got 12 or 11 or 4. If it's got 9, that's exactly 3 codons. And so an open reading frame might be preserved. And it may or may not result in a phenotypic change, a mutation in function. But a lot of times it'll be no good because you've inserted the direct repeat, the other copy of the original target. So when it jumps out, most often it remains mutated. It's very seldom that you can have a complete reversion because it's so unlikely that you'd get that direct repeat precisely cut out. There's not any real good mechanism to do that. It probably involves a recombination event when it happens, but it's not well understood. Rate of reversion, notice how rare that is. 1 in a million to 1 into, 10 billion. Uh-oh, one of these quizzes. No pressure here. What's the name of the sequences that flank the transposase gene? Flank the transposase gene, that means it's a part of the transposon itself. In a simple transposon, it's going to be inverted terminal repeat. The direct repeat would be in the target. I'm hoping it's 2. Please be 2. Yes, it is. Composite transposons. How much? We got about 2 minutes? Composite transposons. There's always a question on composite transposons. So you need to understand how they work. A composite transposon is when you have the same transposon duplicated on either side of something in between from the chromosome. And when it jumps, they don't jump as individual elements. They jump as one big unit. Now, quite often in hospitals and in labs, the thing that's selected for when they jump is antibiotic resistance, because it's easy to select for it. But it could be anything in there. It could be a metabolic pathway, some kind of surface protein, it could be anything. It's just some host genes in here, and they jump as one unit. And the question I always ask, it tests whether you understand why they jump as a unit versus as single independent IS elements. So just think about it. Why do you think they would always jump as a composite? There's something wrong with both of the IS elements. they only have one cut site. The cut site on the outside here and the cut site on the outside there. The internal cut site, the internal inverted repeat, has mutated away so it can't be cut by the transposase. So they can never jump independently, only one side gets cut. But together, it's cut here and here on the outside, and it's taken to the new site. It's not complicated, but that's why they hang together, and that's why it's actually selected for. It's an advantage to make sure these two arms, as they're called, the two IS elements, are mutated on their internal inverted repeats so that it does jump as a unit, because that's the only way you're going to confer antibiotic resistance, or whatever the selection advantage is of having that DNA in between. So it's not a complicated concept. Like most of these, you just need to understand how it works. So you have two IS elements. Of course, they have to be the same IS elements because the transposase has to work on both of them. So the cut site has to be the same. And in fact, quite often, because you have two copies, one gets lazy. You don't really need but one transposase, one gene making transposase. It'll make multiple enzymes. It'll cut here and there. Maybe you'll have a mutated transposase over here, and only one of them is functional. That's quite common. So I might ask you things like, where would you expect mutations to occur or not occur? Well, in terms of transposases, one of them could get mutated like crazy, but you have to have at least one transposase gene functional. Where would you see mutations in the repeats? You certainly see mutations in the internal inverted repeats, but not on the external inverted repeats. Otherwise, it wouldn't function as a composite transposase. And the rest of the slides here will just illustrate what I've told you. Central region could carry, in this case, you have drug resistance, but it can carry just about anything. Flanking sequences can be flip or flop. They themselves can be inverted or not, because it doesn't matter. It's just the inverted repeat that's cut has to be on the outside, no matter what their own orientation with respect to each other is. What's the time? Is it time up? Well, I'm over, and people are not throwing stuff yet? Well, I better quit while I'm ahead. So we'll see you Thursday, and we'll continue this discussion of transposons.